Hi, my name is Bob Fulton. I'm from the McDonnell Genome Institute at Washington University in St. Louis, Missouri. And today I'm going to tell you a little bit about the McDonnell Genome Institute at Washington University here, and the contributions in particular towards the human reference genome. So I'll give you a little bit of history to begin with. So the McDonnell Genome Institute has been here since 1993. I actually started in August of 1994, so I was across the hall prior to that. So I've been around for the existence of the Institute and got a rich history of genomics here. We're one of 4 NHGRI-funded genome centers, along with Baylor College of Medicine, the Broad Institute, and the New York Genome Center. We contributed about 25% of the sequence for the Human Genome Project. We've had about $1.25 billion worth of grants awarded over that period of time. FY19, we had about $27.5 million of sponsored research and a $25 million endowment and a naming gift through the James S. McDonnell Charitable Trust. Just some fun facts to get started. So our throughput in 1993 was about 100 kilobases per day, which was sort of near tops in the world at the time. By 2005, which was the sort of the height of the first gen sequencing, we could do about 200 megabases a day. And today we can do about 18 terabases. So the throughput has changed considerably. 18 terabases is an easy number to say, but it's about 3 times greater than the number of inches between here and the sun, to give you perspective on the number of bases we can generate in a day. 2005 capacity, this is an amazing stat to me, would take 250 years of that capacity in 2005 to equal 1 day of current capacity. So it's incredible the throughput change that has occurred in the span that I've been here. Our data center has about 15 petabytes of storage, or about 30 million laptops worth, and we have about 5 times more storage space than Netflix. We participated in a lot of key projects over the years: Cancer Genome Atlas Project, TCGA, the Human Microbiome Project, 1,000 Genomes. We did a Cancer Genome Initiative years ago. It was 150 genomes, which was an amazing feat at the time. This was in the like 2010. And as well as a Pediatric Cancer Genome Project, and we actually did 2 of those with St. Jude Children's Hospital. We've sequenced a lot of organisms over the years, many popular ones on here. Here in the bottom right corner, I think you can see my mouse, is C. elegans. That was the first genome that we did. We split that effort with the Sanger Institute in Cambridge, England. And we did 50 megabases and they did 50 megabases of the 100 megabase genome, which was published in the mid-90s. And that was actually a nice pilot project for the human, where our director at the time, Bob Waterston, as well as the director of the Sanger Institute, Sir John Sulston, they actually went to Jim Watson and pitched the idea of sequencing the worm as a pilot project for the human. He loved the idea, funded the project, and we used that project to work through a lot of the technology advances that led us into the Human Genome Project. Some other key ones: the MAISE project, top right corner here. So we're not just human, for instance, or animals. The MAISE project was a $30 million initiative through NSF to come up with a MAISE reference. And lots of monkeys, chimps, platypus. Everybody loves the platypus. So for evolutionary studies, so a broad range of things. So in the last couple years, we've been in the process of merging. There were 2 genomics facilities at WashU. There was MGI, which I was part of, and then GTAC was a Genomics Technology Access Center, was another group, and they did a lot of RNA-seq, where we did a lot more DNA sequencing. So we've brought the two together, and we're in the process of moving and remodeling space and all, but we feel like we're getting the best of both worlds. And then we've broadened our institute as well. So MGI in the early days was primarily a sequencing facility. That has now branched out a bit. We have a genome editing, an IPS core now for more functional studies. We have a functional genomics and mutagenesis group for mutagenizing cells and understanding the implications of that mutagenesis. Again, technology development that wraps all around there, and informatics as well. And then again, the GTAC and MGI, which is the genomics facility and the sequencing in particular. We have a lot of omics technologies, from 10x to array technologies, Agilent, Affy, Illumina arrays, DNA sequencing technologies from short read technologies, primarily Illumina on the short read side, to PacBio and Oxford Nanopore on the long read side. We have a BioNano Sapphire for physical mapping, a NanoString to assess primarily RNA transcripts in that instance, a Fluidigm system, which we're actually employing for COVID screening as we speak, and we're hard at work on that. And then a lot of different automation. We use a lot of PerkinElmer cyclones for library construction, Eppendorf 5075s for liquid handling as well, so broad range. So DNA profiling technologies, so a lot of different sources. So we serve the medical school and just the general community as a whole for genomics activities, and we get a wide range of tissues and DNA sources from FFPE, slides, scrolls, cores, blood, fresh frozen, buccal swabs, cell-free DNA, so a broad range of sources. And then we do a variety of manipulations and assessments on those, from chip analysis, DNA methylation, SV detection, again with BioNano or with sequencing, SNP and copy number, next-gen sequencing from whole genomes all the way to a variety of targeted and 10x, as well as high molecular weight DNA applications, which I'll talk a bit more about for the Genome project. In addition, we do a lot of RNA and protein profiling. This has primarily been a GTAC activity that's now merging and we're bringing together. So a lot of RNA sequencing techniques, RT-PCR, expression microarrays, again, the NanoString with the RNA expression, and then SomaScan, proteomics assay. And then, as I mentioned, we have, for short read technology, a lot of Illumina platforms. So NovaSeq 6000s are certainly the workhorse. We primarily run S4 flow cells, and we have 5 instruments on site, and do a variety of read lengths and all depending on the application. But again, primarily S4s for the throughput. All our HiSeqs are now decommissioned. We had 3,000, 4,000 HiSeq X, but we've now decommissioned those for the more efficient NovaSeq, and we still maintain MiSeq and MiniSeq and an iSeq. So then I'm gonna focus primarily, a key project basically that I'd like to talk about is the human reference genome. I've had a great passion for this project for many years. I coordinated a lot of the finishing efforts here at MGI back in the days when we still finished back clones. So spent a lot of time and effort on this project. We've been key participants in the Human Genome Project from the beginning, as I mentioned, our history with the worm and developing the technologies and driving that to where we are today in terms of the human reference sequence. We produced about 25% of the original finished human genome sequence that's in the current Build 38 reference. As well as contributed the alternate alleles, which I'll talk a bit about in a while. We've been a member of the Genome Reference Consortium since its inception, along with the Sanger Institute, NCBI, and EBI. The four groups, we work to basically improve the human reference and add allelic diversity as well, and just maintain the human reference. And we've been funded since 2014 on human reference improvement activities. Prior to that, we still did those activities, but they were more under our large scale NHGRI funding. But in 2014, that sort of split off and became an independent activity that we've continued that throughout our history. We're key leaders in the new Human Pangenome Project, where we're the coordinating center for that effort, as well as a data production site for the project. So the human reference is a work in progress and continues to be and will continue to be. The current reference, GRCH38, is probably not optimal for some regions in the genome and or some individuals or ancestries that you're trying to map to it. GRCH38 is comprised of sequences from many individual humans, so probably over 50 have contributed at least certain regions of the genome, but about 70% is a single individual. And again, 70%, but again, the two alleles from that single individual from the RP11 source. Allelic diversity and structural variation presents many challenges when assembling a representative diploid genome, and we'll talk considerably about that as we evolve into this new pan reference project. Technologies and methods and resources since 2003, when the original finished sequence came out, they've certainly advanced and have allowed for improvements in the reference project and continue to advance at a staggering rate at this point. It's amazing the quality of the data that we're able to produce today compared to the early days. And we recognize that additional high quality reference sequences are needed to represent the full range of genetic diversity in humans, and that's what we're working to explore. So this is a schematic, of the human chromosomes. You see the little red triangles there. Those are regions containing alternate loci. So what's an alternate loci? Basically, it's a region of the genome where we have a different allelic representation sequenced and resolved as well. And these loci are available. So there's a linear chromosomal assembly and a linear reference that's available for download and mapping, and then these alt loci are also available. So they're in Build 38, which was released December 2013. There were 261 alt loci, which comprised about 3.6 megabases of novel sequence. And that included about 150 genes only represented on the alt loci. So there is a fair amount of diversity just captured in those alternative alleles. The average length of those is about 400 kb and the max is about 5 megabases. But there are certainly large regions where there are multiple representations. This, however, presents many problems with mapping. of short reads. If you add these alternate loci, they look like repetitive regions. So you have to account for those. So the software has to deal with that. And this is a slide from Valerie Schneider, who heads up the NCBI effort for the GRC. And so we talk about both haplotypes. It's actually many haplotypes in GRCH38. So samples to be sequenced. So we're trying to sample a diverse range of individuals. And we've, again, been doing this since prior to 2014, but primarily after 2014 when we received funding to do additional genomes. So this is just a schematic of the sort of broad range, trying to capture as much breadth of human diversity as possible. The original sequencing plan, which seems somewhat simplistic at this point, was PacBio reads, de novo assembly, and then trying to scaffold those using 10x and BioNano and Dovetail, sort of binning all that together and putting out an improved genome. But those genomes were sort of a squashed assembly with both alleles squashed together, which today we know is sort of an inferior product in comparison to segregating the two alleles in assembly. So this is just a list of different genomes and their assembly accessions. Some of these are chromosome level assemblies, and some are contig assemblies, but these are all publicly available resources. And again, a broad range of ethnicities. So the process, assembly generation, QC, and submission. So we generate a Falcon assembly, do error correction with Quiver and Pylon. Again, using PacBio reads as well as the Illumina reads to error correct. And these were long read PacBio reads, so high error rates, 10, 12% error rate. We then used BioNano to correct assembly issues and check the integrity of our assemblies and look for chimeric contigs. And then we submit those to NCBI, which were then mirrored to EBI, but to submit those to make them available publicly. Just some general stats just to show, and these will be an interesting comparison to our newer data. But a list of genomes here, the rough genome size based on assembly. Again, remember, these are squashed assemblies, so these are both alleles combined together. They're in 3,000-ish contigs. And the contig N50s range 25, 26 megabases down to, single digits in some cases, 5 and 8. Probably depending on the allelic diversity of the two alleles in the genome, likely contribute to those. Data quality has some implication there, but generally the data quality is pretty consistent. So it's probably more to do with the allelic diversity between the two alleles in an assembly that contribute here. So we use the BioNano again to correct misassemblies. So you see the genomes here, the BioNano conflicts identified, and then manual breaks. So there's 3,000 and some contigs, and we're making somewhere between 50 and 100 breaks in general to correct things. So this gets rid of some egregious errors and puts some obvious things together as well. I'd like to talk a little bit about BioNano and how we use it and how it reveals some of the allelic diversity that we see. So basically, BioNano is a nano-channel device where we put large molecules. Anything 150 kb or smaller is actually filtered out, so we need very large DNA. We do a large DNA prep. We directly label the molecules at specific sites. We run those through the nano channels, and then they stop and freeze those, and they take a picture of the image with the various labels. And then they image the distance and estimate the distance between the label sites. They build a map from that, and then we can compare that to an in silico digest of the human reference to compare the two. So it's basically just a high throughput mapping device. So here's an example. So this is in comparison to GRCH38, which is the green on top. This is Na19240, which is a Yoruban genome. And if you look here, you see two alleles. The blue are the two alleles that have segregated in the assembly. The top one largely matches the reference, where the bottom you see a large inversion. So this is almost like a megabase, 250 kb, something like that, 250, 300 kb of inversion. That again, on a short read sequence and even a long read sequence level, you'd never see this. Additionally, you see some repetitive sequences where this is a repeat that has picked up and flipped over. So this is one thing that the BioNano reveals. And then this is Na12878, so very commonly sequenced genome. And this is the CYP2D6 region. So this is a cytochrome P450 gene responsible for drug metabolism. I think it metabolizes like 50% of the drugs that we take. And interestingly, Na12878 differs from the reference in both alleles substantially. So you can see the top allele here, the red, you see an expansion in allele 1, and then in allele 2 you see another expansion. So the two alleles don't match each other, and neither matches the reference. So understanding this diversity and the implications to the biology are critical. So we have come to the conclusion that we need diploid assemblies, and as the technologies improve, it's possible to separate the alleles. So with increased resolution in high-C data and higher quality long-read data, phasing of the diploid assemblies becomes much easier. We need to develop tools to better utilize the additional allelic assemblies that will soon exist. And that's part of the new efforts here with the pan-genome reference. So thus the human pan-genome effort. So technology shifts. So here's one key technology shift is the PacBio. So a little over a year ago, about a year and a half ago, the Sequel 2 emerged. So the Sequel 1, sort of at the end of its use, these are statistics. We were getting maybe 14 gigabases per smart cell. Reads per smart cell were 500 to 600,000. Polymerase reads 23 kb, insert length 20 kb, and polymerase read N50 of 35 kb. So pretty modest numbers, and these had high error rates also, 12, 13% in some cases. So the Sequel 2 emerged, and the black statistics were as it came out of the box, originally, and then we've had some chemistry improvements that have advanced it significantly more. So the black is beginning use in June of last year, and we were getting about so there's CLR and CCS. So CLR is consensus long read, so basically just generally single pass reads, and we would put as large an insert as we could get to load, and do single pass sequencing. Again, this had the 10, 12, 13% error rate. but we could produce 120 gigabases. With the new chemistry, 150 gigabases. So the throughput was substantially greater, probably tenfold over the Sequel 1. And I thought this was going to win the day with the long reads. We'd have polymerase read lengths at 35 kb and averages in the 20s. So this was pretty impressive data. But then CCS, so consensus, circular consensus sequence. So what circular consensus sequence is we put a circle, well, a linear molecule of about 20 kb or slightly less. We ligate what they call smart bells on each end of the molecule, which then pops open as a circle. And that molecule is then sequenced around and around, and they generate a sequence or a consensus sequence from those circles. So the yield that we get is typically greater than 450 gigabases, but again, some of that is redundant as we go around these circles multiple times. But that typically yields greater than 35 gigabases of Q20 on our recent runs. So over 10x coverage from a human genome in greater than Q20 quality. Typically, the quality is closer to 30 Q30, which I'll show you some statistics from some runs. Reads per smart cell is over 5 million at this point. Polymerase read lengths are large, but again, there's redundancy involved in that, where the insert read length is about 15 to 20 kb, and we're working on methodology to increase that, and I'll talk about that a bit. And again, sub-read length 15 to 20 kb. So NHGRI, with these technology advances, NHGRI announced a new funding opportunity to advance the human reference sequence, which we applied for and successfully obtained. So this is the pan-genome reference, a new beginning. So this is an example of a pan-genome reference where if we look at the sequence here, it goes from blue to red, then you have a green and a purple. So the green and the purple would represent two different alleles. And then again, potential structural differences here at this position. Again, two alternative alleles, and then some common regions. So you have all of these representations, and then what you do is those collapse down into single representations, and then in where there's additional sequences, those are represented as well. So this is the idea behind the pan reference, is to sequence many different individuals and to represent the differences when possible. So the groups participating in this, again, we're the coordinating center. Data productions coming from UCSC, the University of Washington, ourselves, and the Rockefeller University. Assembly from Dana-Farber, as well as other groups around the world at this point. Then Assembly QC, the Sanger Institute, and EBI have been instrumental in that. So some goals for data production, sampling, and diversity is to develop and organize effective networking methods and consortium interactions. Sample collection to ensure full representation of human haplotype diversity. So a lot of population genetics going on here to ensure that we're representing full populations and their allelic diversity. Our goal is to produce at least 350 high-quality reference genomes and produce some finished telomere to telomere genomes. Some goals for the assembly analysis and analysis tools. So assembly evaluation to ensure accuracy and completeness. So there's assembly exercises ongoing now comparing different methodology for accuracy and completeness of those genomes. And then the goal again is to develop a human pan-genome reference and tool ecosystem. So again, this sort of collapsed path where possible, but then unique regions of the genome as well, so that we can assay the common areas as well as these unique bits that crop up in various genomes. And then the goal is to annotate and improve that reference, and then assay any short reads, for instance, that we want to map. We want to map to that pan reference so that we're sampling all of that allelic diversity. For some reason, my slides don't want to advance. There we go. So some outreach goals. So again, dissemination of the results to the community through an effective community outreach program that facilitates a transition to the pan-genome reference. So again, this is sort of a major shift from mapping generally to a linear reference to this pan-genome reference. So this is going to be a challenging effort. And I'm sure any of you who have tried to change references, so we've evolved, the original build for like 34, 35, we're to 38 now. So as we've changed these different builds, it changes coordinate systems, it changes the annotation. So there are certainly complications as you evolve through different references. So that can be very challenging. So our hope is to smooth that out as much as possible. And again, this is a significant change, trying to sample all of these pan references. So we hope to have annual face-to-face meetings with a science day and a symposium. Unfortunately, this year's is going to be virtual, and that's coming up, I believe, in September. Dates to be announced here soon. But again, hopefully we'll get back to face-to-face meetings sooner than later to promote these activities and have open discussions on ways to tackle this. Then workshops at conferences, AGBT, ASHG, Cold Spring Harbor. So ASHG, I know, is probably the next one coming up. It's now online as well. But hopefully we'll have at least a workshop there to discuss on that online. And then targeted discussions with clinical labs aimed at facilitating the adoption of the human pan-genome reference. I think this is one of the more challenging aspects of this, where there are great benefits, I think, to having alternate alleles and mapping to all of this diversity. But in terms of getting consistent, reproducible answers required on the clinical side, there's many challenges with that. Again, I think in many cases it will be more accurate, but there will be more interpretation, which may be complicated. So we certainly have to engage the clinical community to manage this. So our year one goals, so we're winding down in year one, so it ends at the end of July, and we're nearly complete with our first 30 genomes. The aim was to do 30 high quality reference genomes. So our initial 30 came from trios that were all sequenced on Illumina through the 1,000 Genomes project. Again, we are winding down our activities, but PacBio, I think our last genome is actually on the instrument as we speak. So we're optimistic that we will complete all 30, doing at least 30x coverage, greater than Q20 hi-fi reads, insert length somewhere between 15 and 20 KB. Oxford Nanopore, we're using that platform as well, and with a goal of about 60x coverage from 3 flow cells, with average coverage greater than 100 kb read lengths of about 6x. So this is an amazing tool for stitching things together. Again, this has a 7 to 10% error rate. But again, with the incredible average read lengths, it's a very useful tool for stitching assemblies together. And we're using the UCSC Hi-C Omni-C data. So this is chromatin aggregation, basically proximity ligation, and again, phasing almost full chromosome arms in most cases, and helping to sort through the alleles and make sense through of the long read data from an allelic perspective. And then the bio-nano optical maps, again, generating physical maps of the genomes as an orthogonal check. So here at MGI, our current PacBio, I'll walk through the pipeline just a little bit. So we do a QIAGEN MAGATRAC to extract high molecular weight DNA. It's typically 50 kb or greater, which is plenty large for, aiming for 20 kb inserts. We use a megaruptor to shear to a 20 kb shear size. We make smart bell libraries, as I mentioned. We use a Sage ELF to collect fractions, discrete fraction sizes, and I'll talk about all these in a bit. And then the sequencing primer and polymerase, we use a 2-hour pre-extension with a 30-hour movie to collect our data. So this is the initial DNA extraction. Again, we use the MAGATRAC protocol, about 1.5 million cells per reaction for 15 micrograms of DNA. So this application does require a fair amount of DNA. We sheared on the megaruptor, as you can see, and again, you see a broad peak here from 10 kb-ish, 20 kb being the peak, up to 40-some kb. So this is then where the ELF fractions come in. So we make the library first on the full material, do the smart bell ligation, then we do the size fractionation on the ELF. So here, using the ELF 1 kb to 18 kb cassette, we collect fraction 1 and 2. And you can see the difference here where fraction 1, again, it's a pretty tight peak, but it's still large. It's got a large slice up to almost 40 kb, but the primary peak is at 21, but the small end is above 12.5 kb here. Where fraction 2, it goes down in that 12 kb-ish range, but its primary peak is at about 15.5 kb. But again, on the high side, it's not much larger than 20 kb. So there's advantages and disadvantages to those fraction sizes. So this shows just a bit of the impact. So the top stats here, this is fraction 1 and 2 combined, with primarily fraction 1, but some fraction 2 in there. We're getting over 5 million reads, almost 2 million of those get into the CCS and get to a consensus quality of Q20. So we're getting about almost 32 gigabases of Q20 CCS with an average read length of about 16.5 kb and a quality value of Q31. Now, if we compare that to just a pure fraction 1, again, similar yield. The CCS Q20 reads drops a bit, again, because these are longer reads, so fewer of them get around enough times to generate Q20, but the yield is almost 36 kb or gigabases because the average read length is 20.2 kb here. So So you get around the molecules fewer times, so you have fewer total molecules into CCS, but the average length is all over 20 kb. The quality does dip just slightly to Q29, but still a nice high quality value. And then in comparison, the fraction 2, which is the smaller fraction, now we're getting over 6 million reads. So it's a bit easier to load those smaller molecules. We're getting 2.4 million CCS reads. And again, so you're getting around more molecules more times. So you're getting more things into the CCS. Yield, again, is similar to the fraction 1, but the size is considerably smaller, almost 5 kb smaller, but the quality is a bit higher at Q32. So you can see the sort of the impact of the various read lengths. So we're doing size fraction testing with SAGE, the SAGE HLS in particular. So the ELF, we're at sort of the top end of the fraction for the 1 to 18. The next cassette up has given us sort of hit and miss results, and we're interested in pushing the average insert length up a bit as the pack biochemistry speeds up and increases. We're going to need larger inserts to exploit that on the run. So we're trying to push above 20 kb. So we're trying different fractionation methodology to improve on that. These are just sequencing run stats. I think we've largely covered those, so I'm going to skip through this. But then CCS results, this is just another 4 SMRT cells from one of our genomes for the project. So you note here, so these are a bit smaller at 15 to 16 kb average read lengths for the CCS. But again, high quality values at Q31, Q32, and nice yield. So across 4 SMRT cells, we're getting about 140 gigabases of total yield. Here's a bit larger. So these are 18 kb inserts, these fractions. Again, a little bit less CCS yield, a little bit less quality. But again, longer read length. So we're trying to understand the impact on assembly of longer reads with a little less data and a little less quality. So we're trying to better understand what things drive assembly. Here's some coverage titration, 1 through 4 SMRT cells. This is the contig N50, so you can see it increase, but the increase is much more slight from 3 to 4 SMRT cells, for instance. And then this is the number of contigs. And again, the number of contigs does not go down significantly from 2 to 3 to 4. So depending on your application, you can adjust the amount of sequence that you do. And then this is the Falcon unzip assembly results, as courtesy of PacBio. So the primary assembly and then haplotig. So the primary assembly is 1 allele. And again, number of contigs, 1.6 or 1,638 contigs. The contig N50 is almost 30 megabases. The quality value is almost Q50. So again, very highly accurate assembly. This is the haplotigs. So these are the alternate. This is the second allele. And again, pretty good coverage of the second allele. Number of contigs goes up. But again, it has a difficult time when the allelic diversity is low to sort reads. So it's hard to make these full representations, but there's algorithm development to better represent both alleles more effectively. And again, the difference between 4 cells and 3 cells is fairly low. So from there, I'd like to just acknowledge sort of all the efforts. So there's a lot of people that have contributed recently as well as through the years. And many of these people we've collaborated with for years. But you know, the folks at WashU here, NHGRI has been, you know, huge supporters of this effort throughout. And I think they recognize the value of the reference and hope that we can continue, you know, and hopefully they continue to support this. And we're very excited about the new PAN reference. Santa Cruz, UW, NCBI, Sanger, Rockefeller, Harvard, Ithaca, Cornell, so just huge, huge resources here. And then this is an old dated slide, but this is sort of a representation. The names aren't important here. It's really the group at MGI that contribute to all of this, and it's just a great team and a great bunch of people to work with, and we're excited to continue our efforts. So thank you.